Hey, good morning. So I want to welcome all of those here at UAB and the people joining us online to the second week of the Clinical and Translational Science Training Program. Today we're going to focus on Phase 1 and Phase 2 clinical trials. Before we start, I just want to follow up on the comments I gave last week regarding our really brief assignment that you'll all have outside of the class time. So there's a software program called Kaizen, which is something we'll send you instructions how to access. There's efforts underway to turn it into a mobile app that'll be accessible from your phone, but that's not quite ready yet. So this will be something you'll need to use either a desktop or a laptop to access. It's a really great mechanism to provide short answer questions to you to give responses to. The intent of this is to really reinforce the material that you get here in the classes on site. So we will be sending you somewhere between 4 and 6 real brief questions. I don't anticipate it'll take more than 10 minutes to do this. The day after the questions are due, the answers will be emailed to you so that you can see how you did and what the sort of recommended rationale for the answers is. I'm anticipating that this will be ready to be sent by the latest Sunday, and the deadline for completing will be Monday night, and you'll get the answers on Tuesday so that you'll be able to ask any questions the next day when you come to class if anything's not clear. You know, this is obviously, this course is for most people, there are a few people taking this course for credit as an epidemiology course in the School of Public Health, but for most people, this is something you're doing because you want to. And so there's no penalty for not doing the Kaizen questions, although they're really not much work. So I encourage you to do it just to sort of make sure that, check in on yourself, make sure that you got the same message from the presentations that we intended for you to get. You'll also be broken down into small teams for Kaizen, and we will post which teams are leading the pack in the competition for best answers. And if you don't participate, your team will suffer slightly. So I hope you'll take the time to do that. And if you have feedback on how this is working and whether it's worthwhile, I'm happy to hear that. This is the first time we're doing this, but I think you'll find it a useful way to just assure that you're getting the right messages from the sessions that we have here. So today I said the first part of the session is going to be led by Dr. Mansour Saleh, who will be introducing you to the structure of Phase 1 and 2 clinical trials and what they're all about. He's the director of the medical director of the UAB Clinical Trials Unit. He's also the director of the Phase 1 program for the Comprehensive Cancer Center and the Phase 1 program for the Clinical and Translational Science Center. So he has a lot of experience in running clinical trials. He's himself led trials on cancer vaccines, on small molecules that are current cancer therapeutics. He's had a very broad career, both sometime in private practice, doing hematology, oncology, clinical practice. So he knows what the on-the-ground issues are for cancer therapy, and then came back to UAB, I remember, 5 or 6 years ago, to be the director of these clinical trials programs, since he had developed a lot of his own experience in practice as a clinical trialist. So he's a tremendous resource for clinical trials, and really welcome him to kick off this part of the program. To the other side.
Thanks, Dave. So every time I get introduced, I tell the people who introduce me to write that down so that I can use it for my obituary, because it all sounds very good when you're alive. When you're dead, you don't hear it anymore. So I think I should start collecting stuff people say about me so that I can use it at least while I'm alive. Perfect, okay. So I talked about clinical research in general, and phase 1, phase 2, a little bit about phase 3s. But basically to sensitize you about what clinical investigation is, what it means to be a clinical investigator, and whether you're a physician or a basic scientist or a nurse or a nurse practitioner, whatever you are, as long as you're involved in clinical investigation, things that you should be thinking about. So this picture is nearly about 30 years old. 30 years old. And it hasn't changed. You have the clinician there in the middle, who's basically bombarded by patients wanting empathy, the charts to fill out, you could sacrifice, you're going to be away from your children and your family. Some ways the Taj Mahal, the holy grail of publication and academia. On this side, you got to publish, patient care. This looks like Dr. Seth here. He's really the chairman of medicine. He's really making sure that lightning rod's coming your way if you don't pay attention. There's grand rounds, general medicine. I mean, and the VA here, it is exactly, in between, you've got these competitor sharks. I mean, you don't pay attention, somebody eats you up, and you're done. So this hasn't changed. I think it's still the same today as it was 30 years ago when I was a fellow. And then I, who trained in antibody therapy of cancer, where you're trying to give mouse antibodies to patients, and you see here, this is the human land. Well, the mouse gives you an antibody, but you want to cause ADCC, anti-endotypic antibodies. You have immunoconjugates where antibodies are linked to ricin. Ricin is then presented to the tumor cell. But the patient here has diversity. You've got human anti-mouse immune response. How much dose is a lot? Is it too much? Is it too little? Kinetics of the patient in the patient itself. So these issues about the human subject and the device on the other side, and this big gulf of translating what is good in the laboratory to patients. And it is this gulf that still exists today. I think this is what keeps us busy. I think I've learned just as much in my training as I've done while being an investigator, and the issues of clinical investigations remain the same. The complexities become a little bit different, and we'll talk about all of these things. So, well, let me go back. Those of you who come to Alabama, who study in Alabama, have to know about the hallmark of mismanagement of clinical investigation. And you probably just saw the slide. Who knows about the Tuskegee experience? Everybody. Wow. Okay. When I ask, do you think it's a barrier to clinical investigation? Yes. Everybody knows that. Answer is no. When I ask my patients what hinders them from participating in clinical investigation, none of them say Tuskegee, including all the African Americans. In the phase 1 trial, which is toxicity trial, no clinical benefit for the patient, we have about 18% of our patients are African American. The population of African American in Alabama is 26%. But we are doing quite well, 16% African American population, quite well, compared to 26% in the population in Alabama. Yet none of them talk about Tuskegee. What do you think is the biggest barrier to the African American patient or the underserved minority patient for clinical investigation? The top barrier. Tuskegee is number 56. Most of them don't even know about Tuskegee. We're not even born during Tuskegee. What is the most common barrier?
Fear of the unknown.
Probably, yes.
Transportation.
Transportation, childcare. If I could provide transportation and childcare, my accrual of African Americans and minorities would go up to 32%. And why can't I? Transportation, $2, coming and going, $5. Why can't I do that? It's seen upon by the IRB's enticement, inducement. You're paying somebody money, and you may be giving them maybe $4. And maybe they'll give somebody else will give them a ride for $1, they'll make $3 profit. So maybe it's an enticement. And it could be an enticement for people who are impoverished and underprivileged. But so we cannot provide that very readily. But the biggest concern about the barriers to clinical investigation today is not Tuskegee. It is transportation, childcare, those things that make it difficult for a person to be able to give up their daily living to be participating in a clinical trial. So what is Tuskegee? A number of you raise your hands. Tell me, who tells me, what is Tuskegee? What was Tuskegee? You can only be wrong. So be brave and take another strawberry.
It's a trial run by the Public Health Service for 40 years that followed the natural history of patients with syphilis, many of them African American in the rural South. They initially did not have penicillin, wasn't available, wasn't a known treatment in 1932, but it was by 1972. They didn't offer treatment to the patients who were enrolled.
Great.
Followed them.
Okay, full marks for that one. Here, 1932, 600 low-income African-American, all African-American, okay? 400 of whom had syphilis, active syphilis, not informed. And they were monitored for 40 years without treatment, even though in the early 50s penicillin was available. It was stopped in 1973, 32 years. So what's wrong? I got pneumonia. You didn't treat me. It got better. May not have gotten better. Was there anything they could have done to make this be right? Anything. You'll talk fast because I only got an hour and a half, and Bob's here somewhere. He's going to take my time very quickly. Anything they could have done.
No, they were wrong from the start.
Pardon?
They were wrong from the start.
Really? What if I had informed them? You have syphilis, I have no treatment for you, and I would have followed you. I do not know what's going to happen when you have syphilis. I really don't have a treatment for you, period. Can I observe you once a month, draw some blood, and see what's happening? Fair? Anybody? So, informed consent, very important. In HIV, you can ask Mike Sag, we need a randomized, double-blind, placebo-controlled trial in women with HIV in Africa who are pregnant, placebo versus triple agent therapy to protect their child, the unborn child. Placebo control. No different. It's like placebo here. We didn't give them anything. It's like we gave placebo. Well, we informed them. Number one, what's the other criteria that would have made this right?
Provide standard of care.
Provide standard of care? Well, they seem to have gotten it, but they did not get it. Provide standard of care. Third thing, they should be told, if we find something that is of relevance to your livelihood and care, we'll give you that. So for example, I was a PI for study for ITP, thrombocytopenia, where a platelet count of less than 20,000, 25,000 allow you to be on study. It was a randomized controlled trial. Study agent, and the study agent in a phase 1 increased the platelet count in normal subjects up to 400,000. So you come in at 200,000, normal subject, we can increase it to nearly 400,000 up to 1 million. So we knew it worked. It was TPO receptor agonist. It functioned like the thrombopoietin receptor agonist. So we know it does work. So we gave it to patients with ITP who had a platelet count of 25,000, asymptomatic but at risk. Placebo, double blind. So half them got placebo. It was to the fun randomization, so a third got placebo. Fair? I informed them. Unethical? 25,000 patients, you could have a brain bleed. Was it ethical? It was ethical because I provided standard of care. To say if you have a bleed at the moment, I'll give you platelets, I'll give you IgG, I follow you very closely. What else was ethical about it? We said, if you have symptoms and seem to progress on study, I will unblind you and give you the control agent or the study agent, whichever seems to be better. So in this case, we said, okay, we have nothing to offer you. We provide you standard of care, draw blood work on you, observe you. But if something happens that allows you to treat you, we'll do that for you. We'll be totally fair. But we didn't do that. We followed them all the way up to 73 and see what happens in the meantime. Here we are. Tuskegee in the 30s. Nazis in 1940. Yeah, the Jewish experiment by the Nazis going on. Human radiation in the United States of America, testing the atomic bomb in Nevada, okay, to see the effects of the big mushroom cloud. And then Nuremberg. Why Nuremberg is going on? Because of the Jewish experiments by the Nazis. Tuskegee is still ongoing. Nobody talks about it. NIH produces first federal policy for clinical investigation based upon the Nuremberg Code. NIH, oblivious of what's going on in Alabama. The UN talks about it. And then look at here. 1963, the Willowbrook study experiment in a Jewish hospital in New York City. What was that? What was the Jewish experiment on young Jewish kids? Willowbrook experiment. These are mentally retarded children with pneumonia were observed to see what is the effect of pneumonia on children. No therapy provided. This was 63, Tuskegee 1940. We still continue to do the same experiment, no longer on the African American, but now on an impoverished population, a Jewish minority, probably orphan children. Helsinki Accord, 1964. Tuskegee Expose, 1972. Who exposed Tuskegee? Oh, you should know this. This is Trump's, every other tweet talks about this New York Times. Yeah? New York Times reporter comes to Alabama, discovers this, writes about it, becomes a big issue. And then human protection research participants begin here, Tuskegee still ongoing until 1973. As a result of that, Tuskegee stops in 73, Belmont report, etc., etc. Life goes on. Have we learned? You think we have learned from Tuskegee? Yeah, have we? All right, so let's see. We still do human experimentation, exposure to radiation here. And I'll tell you about it in a minute. Here, thalidomide in the 50s. What happened to thalidomide? You know, I went to medical school in Germany. About half of my patients when I was a medical student had no limbs, not all limbs, but half limbs, etc. So I see so often. I went to visit Boehringer Mannheim in Mannheim, Germany, where these experiments were done. What was wrong about that? Why did we have thalidomide in 1950s when Tuskegee was 1930? We already had the Belmont Accord. What happened? Did we inform the women? What was thalidomide used as a sleeping pill for women, anti-nausea pill for women who were pregnant? Yeah. And then 10 years later, 7 years later, because more pregnant women were taking this, they were expected to have deformities of organs. What was wrong? What went wrong in the thalidomide experiment? And how do we correct for it today? We dictate that you use 3 animal models, including 1 human correlate for all your laboratory studies. Those were not used. They used beagle dogs to study thalidomide in their embryogenesis. Beagles do not have the same limb atresia that humans have. When they went back retrospectively, added a new dog species, they were able to see that. So today, the NIH, the NCI, the FDA expects 3 animals with 1 human correlate. Could be a rodent, could be a pig, thus take a pick. Chimpanzee, etc., etc. Now we fight about it. We say, can we do a chimpanzee experiment? We all shout about it. We all protest about it. Well, you got to take your pick. Another subject for another day. So Tuskegee goes on. Look at this. 1999, just about 10 years ago, no, 20 years ago. Gelsinger was a young student who was randomized, or at least offered a clinical trial, died of high ammonia level, failed to disclose this information in the informed consent. There were deaths in monkeys. Not only that, in this, I'm a little bit confused about this. Not only that, there were 2 patients on this particular study who died before Gelsinger received this study drug, but he was not informed. So even today, Tuskegee happens. We see this all the time. I'll give you an example in a minute. We see trouble happening. We talk about it. We say we have to amend the consent form. It's got to go to the IRB. It takes another 6 weeks. What do you do between now and 6 weeks? Where do you document this? And you know, in a phase 1 trial, N equal 12, 14, I put 3 patients on. Somebody in Helsinki puts 3 patients on. Somebody in Germany puts 3 patients on. But there was a bad DLT toxicity in Helsinki. How do I know? Maybe it was a one-off. Should I tell my patient? It's just maybe a fluke. My consent form has not been changed. Should I wait for 6 weeks for the IRB to change the consent form? Or it was a fluke anyway, so you shouldn't change the consent form. So all these things, as an investigator, as a basic scientist who will take your drug to the laboratory and then to the patient, have to think about that. Look at this, 2016, 2 years ago. A phase 1 study looking at a drug that alters mood for patients with anxiety disorder and schizophrenia. It was given to patients. 1 patient left brain dead, 3 patients with irreversible brain damage. What happens? 8 subjects were treated together instead of being staggered. In a phase 1 study, you know, you'll talk about it in a few minutes, you have a 3+3 design. Put 3 patients on, no dose-limiting toxicity, dose level increases. 1 in 3 has a DLT, you add 3 more patients on. Well, but those 3 patients, you know, phase 1 studies are done in multi-center. Those 3 patients go on instantaneously at 3 centers at the same time. Well, if you have a drug which is really toxic, you're concerned about it, you say, okay, let's put 1 no problem, then the next one, and then the next one. Now, that's a problem because you're going to wait for 4 weeks to observe the first patient, then the next patient. You got people lined up here in my phase 1 clinic wanting this drug called Hope. Can I wait that long? Tell this patient, listen, it's going to be 3 months. You're number 3 on the list, but by 3 months in oncology, you'll be dead. And they say, no, I'm willing to take any risk you give me. I'm going to take it. Well, 8 subjects treated at the same time on this study, and then 1 is brain dead, 3 with irreversible brain damage. 5 out of 7 volunteers who were treated were hospitalized, and none of these guys who were put on study thereafter were told about the previous experience.
I'm not familiar with this at all. In phase 1 studies, do the volunteers, do they have to have some sort of terminal illness to even be included in a phase 1 trial?
So we'll talk about that. In my case, it's oncology.
Maybe could you repeat the question?
So the question was, in a phase 1 trial, you have to have a terminal illness to be able to be eligible for a phase 1 trial. Good question. I'll answer it in a minute. So in this case, we had these side effects, we had this toxicity, subsequent patients were not told about it, so people went on expecting to be at least receiving a drug which was safe, but a number of toxicities occurred. 2016 in France. Yeah, could happen in America too. I'll show you an example in a minute. So here we are. UAB just happened 2 weeks ago. So I chair a clinical trials review committee meeting, and every PI comes in there and talks to us about the serious adverse events. So, hyperfractionation is giving high dose radiation therapy in a short amount of time, very focused, to be able to treat a patient who has cancer. Usually, when you give radiation therapy for brain metastasis, you give at least 4 weeks. Small doses over 4 weeks, a little bit higher doses over 2 weeks. Long time. But studies have been done that you can give a higher dose in 5 doses, just as good as 2 weeks as at 4 weeks. So perfect. Let's use the lower dose. Efficacy is just as good. Toxicity may be less. And maybe you can even increase the 5-dose fraction, hyperfractionation, to a dose where you can get better efficacy. So we have a phase 1 study done by John Farfesh. Phase 1 study here. Hyperfractionation therapy patients were receiving increasing doses of radiation over 5 fractions. Standard of care is X gray, which is the dose, times 5, standard of care. So their phase 1 study was increasing the first dose level by 10%. Reasonable, 10%, sounds right. The first dose level was 10% higher than standard of care. Endpoint was outcome at 3 months. So at 3 months, disease control. First 3 patients on study die. What do you ask? These are patients with brain metastasis, metastatic disease to the brain. You were on my committee, and you hear this. John was right there. And what's the question you would ask?
How sick were they?
Yeah.
Start.
Okay, well, the phase 1 requires you to be up and about functional, what we call Karnofsky performance status 0-1. 0 is asymptomatic, 1 is up and about all the time with some symptoms. The question was, how sick were they when they went on to study? Maybe they were dog sick to begin with, so they may have died as a result of just progression. But the clinical trial requires you to be at least well enough, performance status 0-1. What else?
How soon after the dose did they die?
Okay, how soon after the dose did they die? First one died within a week, second one within a month, the other one just before 3 months.
What was the cause of death? Car accident?
Cause of death, always important. Which is why, as a phase 1 investigator, the question is, what is the cause of death? Phase 1 investigator, the question is, toxicity related or unrelated to study agent? Well, first patient died of infection. Immunocompromised host, infection, sepsis, dead. Second patient died, disease progression. Third patient died through disease progression at 3 months. So, all explainable to a certain degree. Okay, now, you are the physician, you want to refer a patient to John Farfesh. What do you say to your patient who's coming to you to say, by the way, you tell them, it's all explainable, immunocompromised host, disease progression, so all very logical. Should you tell the next patient, number 4, something happened? Should you change the study? Is 10% too high? Was it just a fluke? Think about it. This is what keeps me busy. Now, phase one is not just toxicity and just, here we are, give me a low dose, life goes on. I think this is why we have clinical investigation, why we really pay attention to what happens to a patient, which is why physicians, a clinical investigator is not just a good clinician who does this as a hobby. This is an important task. So these questions, because maybe 10% or 20% may be exactly what you need for a patient to have prolongation of survival. So this experiment, very critical, whether you stop it or continue it. So what did we say? We said one thing, inform the patient that the first patient died because of XYZ, inform the IRB, and you may have to change the consent form. But while the IRB is adjudicating all this good stuff, you have to at least document in your chart that you talk to the patient, the 3 patients died, none of them from toxicity of the radiation, as far as we know. You will participate, yes or no? Yeah. Do we do this often? Probably not. Yeah. So what are all these interests? Why do I do clinical investigation? Why am I involved? I love this. I come to work for this. And why? Basically, for the benefit, I keep on saying, it's this mantra, for the benefit of the patient. But that sounds great. Makes me next to God, and I'll go to heaven for sure. Doesn't matter Tuskegee or not. Yeah. But what are my ulterior motives? Academic curiosity. I want to become professor. I need a few papers. I want to publish. Access to novel therapy. I want to tell everybody I've got this great drug that nobody else has. Access to eligible patients, obviously, and you have to do this within the construct of an institution. That's me. Altruistic? Oh, sure. Ulterior motives? Absolutely. So look at this. The NEJM paper requires, in a specific study, 12 patients to be accrued. And at number 11, one more slot for a author left. Somebody else is a patient, I have a patient. You think I'm going to wait on my butt and just let other side put a patient on? No, I'm going to make sure that my patient goes on study. And how do I do this? Whatever you do. Cajole, invite, I don't know what you do many things. So those are all things that I ask myself as a principal investigator as to where I'm altruistic or opportunistic. But that's human nature. Yeah, we are human. What about the institution? What's for them? Achieve academic mission. You heard Dr. Vickers talk about, he gave grand rounds recently somewhere, and he talks about the academic mission. We want to be top 10 in the country, etc. How do you become top 10? So that you have more PIs, doing more studies, more discovery. So we want to really do clinical investigation. On the other hand, they expect you to be compliant, competent. Well, in the context of it, make sure you cover your salary as well. So and it cannot be done with the PI, and the institution really commits to provide every resource for you. But after all, there is also an altruistic mission of the institution, but also an opportunistic mission, because we want to be top 10. Top 10 means high impact journals. High impact journals mean more publications, etc., etc. So we all have some reason to do this for our own benefit. What about the patient? The poor guy is stuck. Yeah, doc, do whatever you want. I'm ready to take it. We'll talk about this phase 1 business I do all. So they are the most vulnerable population, and they could be really the one who would, in a way, manipulate you to do things that you otherwise would not do. I'll give you an example in a minute. And then the sponsor, well, the sponsor. The drug, Herceptin, I was a fellow when we developed Herceptin, not developed, but we worked with Herceptin, anti-HER2. 20% of women who are breast cancer have HER2 positive disease, so anti-HER2 is really beneficial for those women. The cost of HER2 therapy is what? Take a guess. $3,000 per month. In maintenance, it's given for 1 year. So 3 times 12, 36,000. So that was the price when I was treating patients nearly 15 years ago for Herceptin. In the meantime, Herceptin is approved for breast cancer, gastric cancer, and some other cancers. Did the price go down? No. That's how pharma makes money. Money is made by getting an indication, pitching your drug just below the competitive drug, $3,000. And then hoping that the maintenance dose is given for much longer, the longer the better, because we have shareholders. And then when this drug gets approved for another indication, you don't say, I've got 3 indications now, so now I'm going to give it to you for $1,000. You say, no, I'm going to treat you the same amount 3 different diseases at $3,000. Now, I look at it in a very facetious way. It's not that simple. They're going to do more experiments, more R&D, so more investments involved. But that's how pharma makes money, which is why if you play the stock market, you invest in Merck Pharmaceutical, you'll go high very quickly. Last week, watch out because you may go down very quickly as well. I'll give you some examples about that as well. Yeah, but the point to be made is we are all human. We are all altruistic when we become clinicians, physicians, healthcare providers, but we're also very opportunistic. That's human nature. And the question is, what is the balance between the two? How much does it take to develop a drug? If you look at this, we have about 10,000 molecules screened per year. One gets approved by the FDA. A lot of R&D money goes in here. And how long does it take? For a phase 1 all the way up to phase 3 approval, used to be at least 14 years. Too long. Now it's about 7 years. But still, 7 years for one drug out of 10,000 to be approved by the FDA. So you can imagine, pharma really puts in quite a bit of money, which is why Herceptin costs $3,000. CAR T therapy, who knows what is CAR T? Chimeric antigen receptor T cells. So you take your T cells. T cells really will only attack a tumor when they're exposed to small particles of the tumor in the context of MHC class 1 or 2 molecules. CAR T are, you take a CAR T, it's T cell activated, but you put a target on it, like anti-CD19, you'll go straight to CD19 positive cells and attack them. Leukemic cells are CD19 positive, so these T cells that are inactivated in a way will be really targeting the CD19 positive cells, get activated, kill the tumor cell, cure rates are dramatic, nearly 70+%. What does it cost, CAR T cell therapy? You raise your hand, how much does it cost?
Like $300,000.
$352,000, $352, covered by Blue Cross Blue Shield. Expensive, right? So who pays for it? We all pay for it, but this is why it takes this long to be able to screen molecules all the way to FDA approval. Well, it's a business. We have to be sensitive to that. But it's up to us as to how we design trials to be able to make this 14 years become maybe at least half, number one. Number two, the number of patients enrolled in clinical trials in the country today is what percent? What percent of all my cancer patients in this country get enrolled into a clinical trial? 4. At UAB, phase 1, in my own department, 6% of all cancer patients seen by my colleagues are enrolled onto a phase 1 trial. And a phase 1 is, it may be that last hope. The program that I run is called the Drug Called Hope. Everybody comes because they're hopeful that this drug, even though it's a phase 1 drug, toxicity drug, may be beneficial to them. And we'll talk about the answer to your question. 6%. If we double that to 12%, we would cut down how long it takes to be able to approve a trial, and the drugs will become half as cheap or half as expensive. So we can all contribute to the price of a drug by enhancing, or at least being more diligent, but also putting more patients on a clinical trial. So what's a clinical trial? It's an experiment involving human participants who meet predefined criteria for a specific objective so that future patients may use the same drug for benefit. So the difference between clinical trial and just regular patient care is what? Clinical trial is you've got to really meet the criteria on this black box. It's got to be between 18 and 30 or 18 and 50, up and about, functional, liver function tests have to be within normal limits, etc., etc. And then if you abide by those, if you qualify, you can become eligible for the drug X. When the drug X gets approved, you don't worry about that box. You could be 90, I can give you the drug X. So that's clinical medicine. You look at data, experience, and knowledge, and apply. So clinical medicine is application of knowledge gained from research, but then it's up to you how you apply it into what patient population. And the biggest criticism of clinical research is, well, your patient was so well defined, the patients I see at TKC are so different, how can you get the same response rate that you'll be publishing? So that's always the criticism, to be sure that you have to really meet the criteria very well. And when you asked phase 1, and let me come to it in a minute. Let me just jump. Okay, so your pilot study phase 0, where you give a very small amount of drug to the patient, not for toxicity, but just look at kinetics, look at any biologic markers, just a small amount. Phase 1 is toxicity, maximum tolerated dose. Phase 2 is efficacy. Phase 3 is comparative efficacy. And phase 4 is looking at late-term effects. So phase 1 is toxicity, maximum tolerated dose. So it's a drug, never been given to man or woman before, came from the test tube to the rabbit to the chimpanzee, seems to be safe. Thalidomide, I'm going to give it to a patient. Phase 1. You start, how do you start? How do you start a phase 1 study? Just came from the test tube to the rabbit, from the rabbit to a man. What dose should I use? Take a pick. The rabbit, at a dose of a pound, the rabbit died. 10% of rabbits died with a dose of a pound. So the rabbit was about 10 pounds, and my patient is 100 pounds. So you would think that if the rabbit was 100 pounds, it would be 10 pounds that killed the rabbit. So 10% of rabbits were killed by 100 pounds of this drug, 10 pounds of this drug. So what should I give to a patient? So 10 pounds killed 10% of the rabbits. Take a pick. You can't be wrong.
1 pound.
1 pound. Okay, who else? Noel, and we'll talk about it. Let's see. Let me answer this first. So phase 0 is just pilot, pharmacokinetics of an oral drug, often very small amount, subtherapeutic, about 10 patients, and usually often skipped in favor of a phase 1 trial. A phase 1 trial is looking at, it says here, healthy volunteers. And usually in cancer patients, in cancer drugs, you cannot give it to healthy volunteers. So these are cytotoxic drugs that have nausea, vomiting, hair loss, dropping blood counts. You give them to cancer patients. You give them to cancer patients who have failed all other therapies. Your question was, whom do you give this drug to phase 1? A phase 1 drug is given to a patient population who fails therapy approved by the FDA that is curative and shown to have benefit. So in oncology, by the time you've failed 2 prior therapies, everything else is not hocus pocus, but it is really not going to prolong your survival significantly, will have toxicity, and quality of life may benefit somewhat. There's a drug called Tarceva, approved for non-small cell lung cancer. Median survival improvement was 2.6 months. So approved, available, but you say, well, maybe I should go for a phase 1 study because this thing is 2.6 months benefit median. I might as well save it for the future. I'll go to the phase 1 trial. The phase 1 I cannot get every time. I'm getting it now, seems to be targeted. That looks like attractive. Let me do that, and I'll use my standard of care drug later. Allowed. But in general, there'll be patients who really have failed previous therapy for whom no curative therapy is available. In most cases in oncology, if you fail second-line therapy, there's no curative therapy left for you, and most patients will be eligible for a phase 1 trial. We have 44 phase 1 first-in-human trials in my program. 44. Out of which 5 are frontline therapy. Frontline. How could I give phase 1 drugs frontline?
Can you define that? Frontline?
Frontline meaning you come in, you've got cancer, I'm going to give it to you upfront. The 3 other drugs approved by the FDA, they seem to prolong survival, in fact, even improve your cure rate. But my phase 1, never given to man before. Ethical? It's a trick question. Sure, ethical. So phase 1 is dose-limiting toxicity, increasing doses. So what you do is you have a drug combination called FOLFOX. 3 drugs, acronym FOLFOX. Frontline therapy for metastatic colorectal carcinoma, prolonged survival by 4 months, FDA approved. What I do? I take FOLFOX plus a targeted therapy X. It's a phase 1 study. You've never given the 2 combination. FOLFOX remains at standard levels, and the drug X gets escalated. So we have 5 studies today, which are phase 1, first in human, with these are drugs to be given frontline. Never happened when I was training. When I was training, it was at the end of the line, which is what I tell my clinicians. There's no reason why 90% of our cancer patients should not go to a phase 1 trial. On your way to hospice, pass by the phase 1 program. Or the ways to wherever else you go. Why not? There's no harm. Yet, how many patients get referred to phase 1 by my own colleagues who are oncologists who know me well? 20%, 1 in 5. The other 4 in 5 for somewhere else, or don't go anywhere. And out of those 1 in 5, only 1 in 3 qualify for the study because either they're ineligible, come too late, the liver function tests are too abnormal, they cannot fit this particular box. 1 in 5 times 1 in 3 is 1 in 15, 6%. That's what I put on phase 1 studies at UAB Comprehensive Cancer Center. Yeah?
Can I ask you a question?
Yeah.
Do you think of the dose escalation trials as phase 1 or phase 2, comparing 3 doses?
Phase 1.
Phase 1.
Yeah, and we'll talk about it in a minute. So it's dose escalation, 3 + 3 design. Yeah. So phase 2 is you found in phase 1 the dose which was well tolerated or optimally biologic. Either it is well tolerated because it was not very toxic, or if you have a drug, for example, when I was a fellow here, Dr. Bullia was working with, I was Dr. Bullia's fellow, we did the drug called Rituxan. Rituxan anti-CD20. Anti-CD20 is today approved for at least several different indications for lymphoma. It's an antibody to CD20 present on B cells. There was no maximum tolerated dose. You could have given 10 pounds. There was no toxicity in terms of dose-limiting toxicity. So how do you decide how much do I give? 10 pounds, 6 pounds, 2 pounds? How do you decide? Anyone? No hematologist in the room? We have the term called the optimal biologic dose. That dose that blocks all CD20 molecules in the patient's blood cell. Turns out to be 350 milligrams per meter squared, given weekly times 4, blocks your B cells. You'll be B cell depleted for the next 6 months. So no point giving 10 pounds. It's cheaper to make 350 per meter squared. Most people are about 1.6 meters squared. So about, let's say, 700 milligrams. Better to give 700 milligrams times 4 doses than to give 10 pounds, because then you have to make less. So optimal biologic dose, maximum tolerated dose, those are the 2 key criteria for phase 1 determination. Phase 2 is you take the optimal biologic dose, give it to patients for efficacy. And phase 3 then is looking at, okay, I took the 1 pound that was OBD, optimal biologic dose in phase 1, to patients with lung cancer. And the response rate was about 23%. The drug approved by the FDA for lung cancer, for example, docetaxel, is 20%. Is my drug X better? That's a phase 3 study. Comparing docetaxel versus drug X, look at efficacy, comparative efficacy. That's phase 3. Many more patients are required, and Bob will tell you about the statistical design that allows that. Yeah? And phase 4 is long-term evaluation of a drug given for many years on many patients, because you might discover toxicity that otherwise would be unknown. All right, phase 1. Let's talk about this. Let's talk about study. You know, studies have single arm, multiple arms. So one arm gets FOLFOX, the other one gets FOLFOX plus X, the other one gets FOLFOX plus Y. You're comparing FOLFOX plus X versus FOLFOX plus Y. Standard of care is FOLFOX, so you have 3-arm study. Yeah. What is randomization? What is randomization?
Assigning to the treatment group.
How?
Randomly.
Already, right? Flip of a coin, there's a computer that reminds you to say, okay, so there is no bias. Randomization, and Bob will talk about, you overcomes bias. So you don't put the best patients on the drug that you think is going to be the best, i.e. the new drug called X. You give it to them in a randomized fashion. What is blinded?
The patient or the investigator doesn't know which.
Right. So why do we blind the investigator?
Increased bias.
On part of the investigator. Is the investigator biased?
Well, they will be.
Yeah. I'm always biased. I always think that study agent is better than standard of care. So you should really blind me. Why would you blind the patient?
Placebo effect.
Placebo effect? Yes. There's a placebo effect. So we blind both. So who knows? If you blind the patient, you blind the investigator, who is holding the key to this locked box as to what they got. Pardon?
Computer, statistician.
But who owns the computer? Biostatistics, right? The chairman of the board is in the biostatistician. He or she is the one that determines what the patient got, etc., etc. Yeah. What is crossover? When you do crossover, why would you do a crossover? And I think Bob will talk about it. What is crossover? It's not cross-dressing. It's crossover.
Placebo arm to treatment.
Or vice versa, right? You move from arm A. You have 2-arm study. Arm A, if you progress, you go to arm E, or vice versa. And why would you do that?
So that each patient can be its own control.
Pardon?
So that each patient can be its own control.
Right. Yes. Good point. there is a disadvantage of that, doing that, and I will have Bob tell you what is the disadvantage of a crossover. But the study, for example, we did with the thrombopoietin receptor agonist for ITP was a crossover design. Placebo-controlled crossover to say, by the way, if you got placebo and you were progressing, and we know that the study agent already in phase 2 was quite efficacious, I will give you the study agent. So but the trouble of doing that is, at least in cancer, if you look at survival as your endpoint, you cannot determine overall survival based upon your study agent, because now you've got the study agent plus the drug X. You got both. So Bob will tell you about that. What is enrichment? Patients become rich. What is enrichment design? So in targeted therapy of cancer, when you have personalized medicine, where if you are BRCA positive, Angelina Jolie, BRCA positive, we know today that BRCA positive women will reduce their risk of secondary malignancies using tamoxifen by 30%. So enrichment would be, if you have a new drug that works like tamoxifen as a chemopreventive agent for BRCA positive women, you say, fine. Don't take everybody, because tamoxifen has toxicity. Nausea, vomiting, weight gain, uterine cancers, etc., will enrich and only take BRCA positive women, randomize them to tamoxifen plus drug X. So that's enrichment. And there's looking at specific molecular marker that seem to be predictive of response in patients. Yeah. What is stratification? That is my favorite one. And I want Bob to give me the answer, but let's see what you tell me. What is stratification? Bob, what is stratification? Even Bob has to think about this early in the morning.
Well, when you stratify, you're essentially splitting your design in the sense that you're taking into account various characteristics of the patients, in particular demographic or baseline characteristics.
And those are characteristics that may be predictive of outcome. So for example, age may be predictive of outcome. An older person may be less liable to tolerate the full dose, may have more toxicity. So you want to be sure that in a randomized design, you have people who are above 80 randomized, because otherwise arm A might have everybody by fluke above 80. On this side, everybody 25. Well, that study is going to be flawed for sure. So you'll have to stratify to be sure that if age is a predictive marker of outcome or of toxicity, that you stratify. What do you do first? Stratify first or randomize first? What sequence? What? Stratify? Right. Okay, so you stratify first, and then you're okay, fine. You're 80, we'll stratify you, and then you randomize the 80 years old to be either on arm A or arm B. Yeah, okay. Phase 1. So phase 1, we talked about. The basic goal of phase 1 is the optimal biologic dose or the maximum tolerated dose. And you look at this dose called MELD-10 or NOEL. And what you do is you dose escalate, not in the same patient, but in patient groups. And you have usually small increments of escalation. So what you do is, NOEL is no observed adverse event level. NOEL. And MELD-10 is murine equivalent of lethal dose in 10% of animals. And then you have the human equivalent dose, which is HED. So in a phase 1 trial, you take a 1/10 of MELD or a 1/10 of NOEL. So NOEL is no observed elements, no observed toxicity. You take a 1/10 of that, really a low dose. Or you have that dose that killed 10% of animals, you take a 1/10 of that. So 100% lethal for the mouse. You take it in the human equivalent dose. So these are at least some approximation markers as to how you start with the drug. On the other hand, if you already had a monoclonal antibody anti-CD20 that binds to CD20 molecules, if the approved drug dose is 350 milligrams per meter squared, QE times 4, well, the next anti-CD20 would be, you won't start at NOEL 1/10 of a 10th. You would say, might as well, you already know 350 is good, I'm going to start at 150. In my animal model, the PKs are very similar to Rituxan and CD20. In the human correlate model, it's similar, so I have to start with 10. I have to start with 150. Yeah, David.
I want to be sure I understand what MEL-10 really means. So a mouse is 20 grams roughly. So are you saying if a milligram was lethal in 10% of mice, you would give 1 milligram to a person, or you would give 1 milligram times the multiplier to go from 20 grams per mouse up to a 70 kilogram person.
So you take the human equivalent dose of the 1 milligram in a mouse to the human correlate. So if a mouse, and there are some calculations to say if you use a milligram in a mouse for the human, based upon the kinetics,
Right.
not kinetic, but blood volume, etc., it'll be maybe about 100 grams. So you would use 100 grams as your MELD 10, and then you use a tenth of that for the first dose level for a patient.
It seems like there might be some drugs where 1/10 of the 10% lethal dose in a mouse might still be 5% lethal in the mice. And that would be important information to know, because you wouldn't want to do, I mean, it would be really different. I guess the assumption is that 1/10 of a lethal dose is going to be 0% lethality in a mouse.
Man, you mean?
Well, in the mice. So if 1 milligram is lethal to 10% of mice, 0.1 milligrams might be lethal to 5% of the mice, or it could be lethal to 0%. And it would be important to know the difference between those to decide what dose you would ultimately use.
So let me ask Bob, who's sitting next to you, as to, he's the one who usually helps us determine what the starting dose should be. And I have an answer to that, but Bob, do you have a thought about that?
Well, to determine what the actual starting dose is.
Yes.
I mean, I help with designing the study itself, and when dose escalation should be or should not be, and so on. But I mean, I don't determine what that is.
So he's taking the fifth. So let me tell you, which is fair. I think all of us are guided by L or MELD 10. On the other hand, we look at the molecule itself. If the molecule is an antibody, unlikely that the molecule of the antibody will be lethal. It's binding to CD20. The only lethality would be you really deplete your B cells. So say, fine, I'll start with MELD 10, a tenth of MELD 10. If the molecule is blocking a specific pathway in a patient, a pathway, for example, EGFR, critical in patient homeostasis, we say, well, never given to men before, MELD 10% of that would be a milligram. Let's be safe. Let's take 0.5 milligram or a tenth. But you're right. I think, which is phase 1. Is it possible to have lethality in phase 1? We saw the trial that John Fifish did, right? So I think it's fair to say that in phase 1, the patient does take a risk, although lethality today in phase 1 is less than 0.001% in human studies. Because we've learned, it may well be that we really go to MELD, not just a tenth of a tenth, but 100th of a tenth. The trouble is, if you go lower you go to put more patients on study. So I tell myself, if I was a patient, as a clinician, when I see patients, I think the guiding principle for most of us is, why them, why not me, and when me? Universal question. When me? And if it was me, would I want to be in their shoes as I'm treating them? And that makes me really grounded to what I would do. So I tell myself, and I tell my patient as well, you don't want to be the first dose level in a patient study, phase 1. It's the lowest of the low dose. In my curriculum, and I don't know whether I can say this online, I say it's like peeing in the ocean. You will not change the color of the ocean. Too low. You don't want to be the last patient or the last dose level. It's going to be toxic for sure. You want to be somewhere in the middle. But you can't choose when you get your cancer. So there is clearly much altruism on part of the patient who are willing to come on a phase 1 study, or any study for that matter. It's a drug called hope, yes. There's also the ability to say, I want it to help me. I really want to try if it works for me. If it doesn't, it'll work for somebody else. But those are the constellations of what goes on in the patient's mind. It's up to you to provide the most informed information and to be really clear when you have informed consent. So phase 1 study, dose escalation, you put 3 patients, 3 plus 3 design is 3 patients, 1 dose limiting toxicity, and you determine what the DLT should be. For example, in a small molecule that blocks a pathway, EGFR pathway, the DLT could be increased liver function test because it is metabolized by the liver. The DLT in a CD20 study, anti-CD20 would be B-cell depletion. If the B-cell are depleted really significantly, well, you don't want to escalate the dose any further. You are 0. No B-cells left for the next 6 months. So that's the optimal biologic dose. So your dose-limiting toxicity is often defined by the toxicity of the drug, and then you define that as a DLT. All other side effects are not DLTs. So for example, phase 1, 3 patients on, you put 3 patients on, and the DLT is confined to the first dose level. First dose level is usually about a month. You give a drug once a day, every day for a month, or one dose every week, 2 weeks on, 1 week off, 1 month is a cycle. We only define the DLT in a month's period. So if the same toxicity, liver function test abnormality, occurred in the second cycle, the second month does not count to the DLT. Now there's a flaw in there in somewhere because you have dose accumulation over time, but then you also have toxicity of the drug accumulation, but also you have disease progression. So by convention, we usually just have definition of DLT based upon the activity of the drug and how it's metabolized and the first cycle. Now, if you see many patients experiencing DLT in terms of toxicity, but not DLT specifically, in the second and third cycle, you have to go back and say, by the way, something's wrong. There's drug accumulation. Let's go back and look at the PKs and then go and redefine whether you go back at the same dose level or you go to a reduced dose level. But in general, at least in oncology and most other specialties, the DLT is defined by the first dose level. So you put 3 patients on, no toxicity, you go up. So you go up by how much? So we start with 1/10 of a tenth of MELD, 1 milligram. No problemo. 3 patients done, no trouble. What's the next dose level? 10 milligram, 50 milligram? So the statisticians give us some guidance. They have this business called Fibonacci. I like the term. What is Fibonacci, Bob? The Fibonacci sequence of dose escalation.
Well, it's just a sequence based on probabilities that can be calculated that used to set up the dose escalation. I mean, I don't really want to go into any details right now.
Is it magical?
Oh, no, it's not a magical thing. Not at all.
So we have Fibonacci dose escalation. You just double the dose. So it's basically a guesstimate, an educated guesstimate. We work with a statistician to determine. The advantage of rapid dose escalation is what? You get to your DLT, MTD very quickly. Study's done. You can go to a phase 2. If you don't do rapid escalation, it'll take a long while for you to get to your DLT, MTD phase 2 dose. On the other hand, if you do it too rapidly, you really suffer toxicities and your drug is dead. So you've got to find the middle of the road. So we work with the statistician, work with the clinician, what are the potential toxicities, and determine what your dose escalation is. And there are some statistical methodologies to allow you to do that, Fibonacci and others. Yeah. So 3+3 design, you put 3 patients on, and if there's 1 DLT, you put 3 more, because 1 in 3 is 30%. 30% could have been just by chance, could have been just a fluke. So you put 3 more, so it becomes 6. If 1 in 6 has a DLT, it's 15%. We will not escalate above that. So most of the time, a phase 2 dose level is that dose level that has 1 or less DLTs in 6 patients. And you can change it each whichever you want to, but most people would say, I wouldn't go above 1 in 6 as a dose-limiting toxicity. Sorry, go ahead.
Does this not apply to an agent that's used already for treating vascular disease and you want to treat the new disease? So, like for example, I'm looking at a drug that's used for rehabilitation and I want to use it for another indication. So I know the dose that can treat this condition and the toxicity. So do I have to go through all of those phases, or can you use the equivalent dose of for the first indication? Like say for malignancy, if you use this approved for certain type of malignancy and use it for another type, what are the steps that you have to go through?
So the question being asked is, if you have a drug of the same class, the same molecular constellation that blocks the same pathway, and this drug already been approved, anti-CD20 approved, Rituxan by Genentech, does the next anti-CD20 have to go through the same process? And the answer is yes. But you may not have to go from MELD 1/10 of a tenth. You could start in the middle, but you step through a 3+3 dose design, or there are other designs, and I'll show you in a minute. You have rapid escalation where n equal 1. You don't put 3 patients, you just put 1 patient. No toxicity, you go to 1 more. And then when you start seeing toxicity, then you do a 3+3 design. You have a rapid escalation. So you're allowed to do that, but you have to go to phase 1, phase 2, etc. And that becomes more complicated for biosimilar molecules, which are biosimilar, work similarly, but are synthetically generated, and you don't have to go through the whole process of DLT, MTD, but phase 1, phase 2, phase 3 have to be done.
What if the same exact drug is used though for a different indications.
point.
You already have the clinical toxicity trial data, and you want to take drug A that's already approved to treat disease A, and you want to see if it treats disease B. Can you just jump to phase 2?
Yes, you can jump to phase 2. Phase 1 is done. Now, you might see a phase 1B to say, you use rheumatoid arthritis, anti-IL-6. But it could also be used for sarcoidosis. But sarcoidosis has a problem with lungs, and this drug has some toxicity for the lungs. You do a phase 1B. You look at one dose level below the MTD. 3 patients, no problem. 3 more at the MTD level, done, you move on. So at least for your own safety, because there may be cross-reactive toxicity between the drug's toxicity and the patient's biology of the disease, then I would do that, phase 1B.
In a case where you have a drug that is used to treat, say, high blood pressure, but the clinicians in the clinic notice that it also is, I don't know, doing something else, so it's like almost a beneficial side effect, then do they still have to go back to phase 1 trials, even though we know the benefit is there?
Well, it goes back to the question, do you have to go all the way to phase 1? Phase 1 is toxicity, so you already know the toxicity of the drug, what dose is well tolerated by the patient, so you will go to 1B. Example, IL-2, interferon. Interferon was used for melanoma, and in the context of treating melanoma patients in phase 1, phase 2s, and phase 3s, we realized that men and women have long lashes, eyelashes. Great discovery. But on the other hand, it had been used by Revlon very quickly, but there was significant toxicity to interferon. But there was a study done, a 1B, and a small phase 2 to say whether it was used for eyelashes and a slightly lower dose, same dose, but a different interval. But you can do it, but I would do a phase 1B.
Okay.
What was the time? 9 o'clock? Half an hour left? Okay. I have this watch that only functions when it drives itself when I shake my hand. But when I shake my right hand, it doesn't do it on my left hand. So I got to work with both hands. This thing is stuck at 8 o'clock. I got to move my right hand, my left hand. All right. So dose escalation, increasing doses. There's also a pharmacologically guided. So you look at pharmacology, you look at your design, and then you look at your PKs, the area under the curve, and that determines how quickly you can escalate. And then you have these very novel statistical methods, and Bob may talk about it, to say you look at the hypothesis, then look at toxicity, and how frequently the toxicity has occurred so far in the first 3 patients. And then you decide, based upon that, and the predictive value of the toxicity seen before in other patients with similar molecules, you may jump to 10 times more at the next dose level. So all kinds of things that the statisticians work with to determine. The earlier you get to a phase 2 dose, the better you are. But the trouble is, it's like thalidomide. Now, thalidomide was a problem because we used the wrong animal model. But very often we've gone to phase 1 very quickly, got a DLT, MTD, optimal biologic dose, done a phase 2, and now you're stuck because you have toxicity you did not anticipate because you're too much in a hurry. It kills the drug. Yeah, so you have to be very careful. That's what pharmacology and the pharmaceutical company really worries about. Okay, here. Now this is going to make you think. So we have a drug developed here by Dr. Chowdhury in the Department of Surgery. A milligram of this drug significantly extended survival in rats and pigs subjected to 16% controlled hemorrhage. That means if you bleed the pig down to 40% of the hematocrit, all pigs die. On the other hand, if you give 1 milligram of this good stuff, the survival was 6 times higher for study agent versus vehicle. 80% of animals at the 1 milligram per kilogram group remained alive for 6 hours. No animal on the placebo group survived. And out of those 80%, half, 40% survived forever after. So 1 dose of a milligram per kilogram of E3SO4, it's an estrogen derivative, survived 6 hours. Half of them survive forever after. So the army loves this drug. They say, by the way, it could be used in warfare. You get blown up by some shrapnel, some bomb. You are about to lose your life. 1 shot of this will at least keep you surviving for 6 hours, 80%, get you to the medic, get you to the next hospital, you're done. So, in fact, there's this phenomenal, we saw a colonel come here and talk to us about it. The future warfare will be run by robots. So if you fall in front war somewhere in Iraq, there'll be a drone, they'll land right where you are, intubate you, put an arterial line and a venous access and give you a drug of a dose of E3SO4. All done. All tested out. Well, be ready for it. You thought playing drones just to take pictures was easy. That's going to be more complicated. Okay, so phase 1 study. The one thing about this drug is it only works in a hemorrhagic state, in the hypovolemic hemorrhagic state. So efficacy will have to be done in that particular setting. So we have a phase 1 cooking, about to cook, once we have these animal studies done, and we'll do a phase 1 in healthy subjects. Escalation, just look at PKs, toxicity. It's an estrogen derivative, so you would think that men would get hot flashes, women will get more hot flashes, change in menstrual cycles, simple stuff. Not quite simple, but simple. Yes.
How do you get informed consent in a case like that where you have somebody coming in that's hemorrhaging all over the place?
I'm getting to that. You should give my talk next time. So I'm getting to that. So phase 1, you can take all comers, right? Healthy subjects. As an oncologist, I wrote the phase 1 study. We'll say we'll do it in healthy subjects, all the way in the campus. And Guru here signed up already because we're giving $1,000. So Guru said, I'm going to sign up for it. This was 3 years ago. So he hasn't gotten his money yet because the study is stuck at animal water stage. What about women? Women take birth control pills, also an estrogen. Should we use women? Well, we said no, women, because it will interfere. This is a synthetic estrogen. But the Army said we have half of our Army people are women. So we'll do the women in the phase 2 part. So phase 1, men only. They sort it out. They realize what it is to have hot flashes. So we done, well, let's see, assume 10 milligrams per kilogram, 1 dose is the optimal biologic dose. It blocks the adrenergic receptors, whatever receptors are involved. Phase 2. Phase 2 is going to be in patients who are hemorrhagic or at least hypovolemic. Question is, do you do it in a GI patient? How do you do it? So let's talk about a phase 1b. I want to do a phase 1b before I go and put up 100 patients in a phase 2 trial. I know that this drug only works in the hypovolemic state. These rats, rabbits, pigs were bled all the way down to 40% of hematocrit. Tell me, what design? Can it be healthy subject or could it be ER patients? The trouble about hemorrhagic patients who are undergoing a GI bleed is they're going to the OR to get things done. Everything's splenectomy because they ruptured their spleen. I mean, there's no way to get kinetics. Their outcome, survival, is based upon the great surgeon and this poor drug. The drug is already bled out as the guy's bleeding, so it's all in the drain somewhere. So difficult. Consent form, yes. The guy is unconscious. There is waiver of informed consent, where you waive consent because the drug is not toxic. You can give it to the patient based upon the ethical responsibility of the PI available. So all ER studies done in by the ambulances when you are in a car wreck are done waiver of consent because you don't have next of kin, which is the next thing. Next of kin gives you consent, but if you don't have a next of kin because you're in on the highway somewhere, there's waiver of consent, but only in studies where the drug itself does not have significant toxicity or lethality. Yes.
But can you say that about this drug before it's even gone through phase?
No, well, what I'm supposing is it's already going to phase 1. Okay, you got a dose of 10 milligrams per kilogram, optimal biologic dose. It sustains the patient's, the healthy subject's blood pressure, whatever it is. There's a marker. I'm doing a phase 1b now. In phase 1b, in healthy subjects, we cannot go to hemorrhagic patients because it's too uncontrolled a setting. If we have a good outcome in phase 1b in healthy subjects, then we can go to phase 2 in ER patients, hemorrhagic or up there. By that time, we'll know what the toxicity is. But I'm asking you, in healthy subjects, what's a constellation of patients I would design to be able to test out this drug in the hypovolemic state?
I had a question. Assuming you've tested it in healthy patients for toxicity study, what is the purpose then of the 1B if you need that before you go to hemorrhagic patients?
Right. 1B is to say, by the way, efficacious in pigs. There's any biologic activity, but you got the toxicity already in phase 1. Phase 1B is optimal biology, i.e., can you really raise the blood pressure of a hemorrhagic patient without being hemorrhaged, i.e. in the ER? So there, 1B is look at biomarkers. What are biomarkers of success? The biomarkers of success that we saw in animal model increased nitric oxide, etc., etc. So the 1B is look at those biologic markers to say, yes, this dose will not bring the blood pressure up because it's difficult to bring the blood pressure up. We couldn't hemorrhage the guys that low, but at least the nitric oxide went up, i.e., okay, there's some biologic evidence of activity. And then you say this drug can go into 100 patients phase 2. So my question still is, in the healthy subjects, how can you mimic the hemorrhagic state? You tell me the answer, I'll let you go, and you can have half an hour free. Pun?
Could you really ethically though? Would you have to give them something?
Those who donate blood.
Pun?
Those who donate blood, like
They donate 1 unit. 1 unit is
Donate 1 unit.
10%. 10% is too little. So you could say, we donate blood all the time. The people all the time. Most of us do not do it, but we do. Donate blood, 1 unit, 500 ml. 500 ml is 10%. 10% is too low. You got to go higher. So
People who might need a transfusion, but aren't so low that they're hemorrhaging.
yeah, so many ideas. Long story short, so what we decided was, and we won the competition, we said, okay, we're going to get at least a 20% hemorrhagic state. We can't get a 60. 60 is lethal. 20% hemorrhagic state, and then we'll have biomarkers, nitric oxide, there are a number of things. We was helped with the study design, a number of biologic markers that tell you that this drug is working the way it's supposed to work. Stops capillary leak, etc., etc. We said, okay, let's at least get a 20% hemorrhagic state. So we talked to the blood bank, talked to the RB, all done. We phlebotomized you 2 units. 2 units is 1,000 cc's, 1 liter. Everybody, when phlebotomized by 1 liter, will be dizzy, tachycardic, you're sweating it out. It's okay. We phlebotomize you 2 units, keep you on a bed stretcher, give you the drug, observe you for 6 hours to get all biologic markers, impediment observation, blood pressure, tachycardia, nitric oxide, etc., etc. At 6 hours before we send you home, we give you back 1 unit. So now you're -1. The same way you would be if you were giving blood at the blood bank. So I thought it was a smart design. -2 + 1 becomes -1, you go home. But still, it is going to be a little bit tricky to say, by the way, -2, can you really sustain yourself that long for an hour, 6 hours for PKs? Will you get dizzy? You'll overlook somebody's heart disease. Some young student who wanted $1,000 has a valve disorder, and all of a sudden gets significant toxicity. So all those things come into play when you do that in healthy subjects. In the cancer patients, the risk is much lower. The patient's willing because they have a lethal disease. A young subject is totally healthy, Gelsinger, yeah? Okay, so that's what we did, and we're waiting for this drug to get FDA approved. Phase 2, as I told you, efficacy. You look at the phase 1 optimal biologic dose, and phase 3 is comparative efficacy. But I want to get to something else. Bob might talk about. There's this 2-step design. So I have a drug in phase 1, 1 pound, not toxic, does well. Phase 2, usually about 20 to 40 patients. But it could be that this drug may be a dud. Am I going to give it to 20 patients or 40 patients just like that? And what's the cost of a study patient for a phase 2 study? How much does it cost pharma to give me to do a phase 2? Let's do that, bro. How much does it cost pharma to give me to do a phase 1 study per patient? What do you think? How much does it cost? Genentech, they pay me how much per patient, 1 patient?
They pay you? Or
Yes, they pay me because I've got to do the study, right? I've got to do PKs, CT scan, not pay me in my pocket, but to pay for a CT scan, assessment, physician visit, the entire design of a 6-month study, the phase 1. Yeah? So they pay the institution. Just take a guess. $40,000. $40,000. Phase 2, about $30,000. So imagine 40 patients times $30,000, phase 2, a lot of money. You don't do that and realize that this drug was a dud because the phase 1 gave you at least the MPD, OBD, but now is really efficacious. So we have this 2-step design, and hopefully Bob will talk about it. You do a 1-step, i.e. put 14 patients on. If no response is observed, the drug is considered not likely to have a response rate of 20%. How did that happen? That is, failure 14 times in a row would happen only 5% of the time if the drug truly had a response rate of 20%. Very complicated. 0 out of 14, 1 out of 14 is, to me, 6%. I don't know how we got to 20%, but the Bobs of the world will say, if 14 times in a row you get a dud, that drug is unlikely to have a response rate of 5%. So we say 2-step design. We do 1 step, 14 patients, 0 of 14, shut the study. 1 of 14, there is a likelihood that it could be 20% responsive. You put at least another 10 to 20 patients, and maybe Bob will talk about that. Yeah, so we have two-step design for a phase 2 study. And then phase 3 is hundreds of patients to look at efficacy. There's also a randomized phase 2, and Bob may talk about it. The endpoints, there's a survival endpoint, disease-free survival endpoint, time to progression endpoint. There are all these endpoints in oncology that may not be relevant to non-oncology. But I want to get to something to explain to you. How much more do I have? 4 minutes, okay. So I want to try to let you go home understanding the responsibility that you have as a clinician, that we clinicians have in our design. So T cells only work when the tumor is lysed by radiation chemotherapy, where it is you have small target antigens. They are picked up by the dendritic cell in the context of a MHC class. That T cell, that dendritic cell, then presents it to the cytotoxic T cell, and this naive T cells become activated and then looks upon the tumor as foreign. So it requires T cell target antigen, the dendritic cell, which is the antigen presenting cell in the context of MHC, T cell, which is inactive, becomes activated, and this activated T cell will then be anti-tumor. That's the philosophy of T cell activation. Different from B cells. B cells get activated instantaneously by binding to the B cell receptor. So what happens? Here's a dendritic cell, the activated T cell, presents the antigen to, the other way around. Here's the tumor cell. Here's a dendritic cell. So yes, the dendritic cell presents this target antigen in the context of a T cell receptor to this tumor cell in this particular context. And then they have PDL1, PDL2, PD1, PD2. These are receptors, and we have ligands. The T cells gets inhibited when these two receptors and ligands mate. So when you have activation of the T cell, after being presented by this antigen, and now becomes anti-tumor, both the tumor and the T cell have these PD1, PDL1 interaction, PD2, PDL2 interaction that inactivates the T cell. So if this, you get ligand receptor linkage of PDL1, PD1, or PDL2, PD2, this leads to inactivation, senescence of the T cell. So if I told you, I want to activate the T cell, this is what happens instantaneously after the T cell has the antigen presented to itself. Here. So the antigen was presented in the context of T cell receptor, etc. This T cell got activated, but this constellation will inactivate the T cell. And I want to activate the T cell. I want to block this particular process. What would you do? $1 million question. Too easy. You want to block this interaction. How do you block the interaction between ligand and receptor? No? Pardon?
Antibody that can block the receptor.
Sounds good. You should offer Genentech. Here it is. Approved by Pharma, Keytruda, anti-PD-1. There are 6 anti-PD-1s available. There are a few anti-PD-2s available. They block the interaction between PD-1 and PDL1. Now the T cell gets activated, and that T cell activation leads to anti-tumor activity. So, study done. Nivolumab, anti-PD-1, in patients with non-small cell lung cancer who have failed 1 prior therapy. So the first prior therapy for lung cancer is carboplatin and Taxotere, for example. The next therapy after you fail frontline is docetaxel. So there was a randomized trial to look at this molecule, T cell activating molecule, anti-PD-1 nivolumab versus docetaxel. Phase 3 study, nearly 300 patients each. Okay, look at this. Here, this is anti-PD-1. This is docetaxel, clearly superior, right? Everybody can see superior. So the question is, what characteristics? And maybe Bob will talk about it, but look at the forest plot. If you look at this is positive for nivolumab, this is positive for docetaxel. Everybody, in terms of overall survival and progression-free survival, if you are PDL1 positive more than 1%, you had benefit. More than 5%, even more benefit. And if you're more than 10%, even more benefit. So you had to be at least 1% or more PD-L1 expressing on your tumor. Yeah. If you look at the other way around, overall response rate, nivolumab positive here. Patients who are more than 1% positive for PD-L1 had a better response than if you were negative, and 5% was even better, and 10% was even better. So it was clear that this drug works only in the context of the tumor being PD-L1 positive 1% at least. 0% was useless. So here we are. If you look at this, more than 1%, less than 1%. Every time you were more than 1% or more than 5% or more than 10%, there was superiority of anti-PD-1 compared to if you were less than that. So the drug code approved for a response rate benefit, overall survival benefit, overall survival 12.2 months, median versus 9.4 months, response rate 19% versus 12% in patients who are PD-L1 1% positive. Drug code approved in the second line setting. All right, last question to you. So you're the big boss of the pharma. The drug just got approved for every patient 1% or more in the second line setting. You want to give it in the first line setting because that's where all the patients are. The second line setting, you only get a third of the patients because a third die. The other third are ineligible. You only get a third. You want to make big bucks. You want to get everybody who's got lung cancer. What's your study design? Non-small cell lung cancer, I give you everything else. The guy's healthy as a horse, like Schwarzenegger, good performance status, good laboratory functions. He is the best you can be. What's your study design? You're giving, once again, standard of care is carbotaxol. Give it to you already. Then we'll use nivolumab. It was already approved in the second line setting for 1% plus positivity. So give me at least one criteria that will be very important for the phase 3 study, knowing what I just told you. Pardon?
More effective than the baseline.
Sure, but that is your objective. You want to be more, so objective is you want to have your study agent be at least 20% better in overall outcome, or survival, or response rate than study agent. Yes, but the question is, for the eligibility criteria for the study, what do you want the patient to be? It must be PD-L1 positive. How much?
More than 1%.
More than 1%. Give me a number. So you would accept the same criteria as the previous study as more than 1%. Yeah, more than 1%. Going once, who else?
More than 10%.
More than 10%. Why?
Is it
Was more efficacious than 1% on the forest plot. Wasn't designed that way, but it was at least seemed to be like that. You're all investigators. You could ask me a question.
Then your results are less generalizable because only a small proportion of the population has 10%.
Sure. So you want to be greedy and be everybody who is more than 1%. That's being greedy in a way.
Well, it's not greedy. I think it's extending your therapy to a larger population of patients.
Okay, got it. All right. So 1%. Somebody says 10%. Anybody else? You're going to ask me a question. The question is this one. Look at this. This was a study, docetaxel, anti-PD-1. Progression-free survival. This is, where are we? Docetaxel is gray bar. Gray bar, if you were 1%, you were equal. Okay? This is 1% or less. This is 25% positivity, much higher than docetaxel. 50% even more higher, and 75% even more higher. So with this data, the higher you are, the better you are. Sure, 1% and more gives you a big catchment. 75% gives you a smaller catchment, but the chance of this working is dramatic. So now I'm asking you, where is your cutoff? Take a pick. Be brave. Either lose money or make big bucks. So I'll tell you, two companies went in this way. One company said it'll be 1% or higher. The other one says, no, we're going to be conservative, 50% or higher. The treatment of choice, frontline therapy today for non-small cell lung cancer is nivolumab or Keytruda for patients who are 50% or more, because 1% or more was a negative study, was no better than chemotherapy. That is the power of statistical methodology, study design. You want to be encompassing, there's a risk. It was better in the second line setting, 1% or more, But in the frontline setting, you're giving 2 drugs, carbotaxol, not docetaxel. They have a better response than docetaxel alone. So you're going to waive that out. So it turned out that the 1% plus study was negative, and those people are 30% positive, 50% positive or positive. So today, if you are lung cancer, non-small cell, frontline, if your tumor is 50% positive for PD-L1, treatment of choice is non-chemotherapy. You keep your hair, you get anti-PD-1. That's what keeps us awake, alert, data-driven. But yeah, it's a guessing game. You can imagine billions of dollars were lost by that one company that lost to the other company. Now the catchment is very short, small. Well, better off that way. You can still then sneak down. What about 25%, et cetera, et cetera. Yeah. Okay, I got 7 more minutes. 7 minutes? Okay. 2 minutes. Of course, it's 2 minutes. But you can ask me a question before I let Bob talk, because statistician, I think the best partner for the patient is for the physician is the patient and the statistician. They always are quiet people behind the scene, but their methodology is very critical to you being able to achieve your outcome. I'll entertain 1 or 2 questions, and then I'll give over to Bob.
Okay,
All right, thank you.
great. So we'll take a 2-minute break, and then we need to move on. So if you need a water break, take a moment. Thanks very much.
Thanks.
For your help.
Really? I've been calling it work for years.
Yeah. Thank you so much. Good. Just one thing.
All right, well, go ahead and start. I'll be essentially just.
I'm going to just introduce you really
Sorry
briefly.
about that.
That's okay. The last part of today's session will be led by Dr. Bob Oster. He's a professor in the Department of Medicine in the Division of Preventive Medicine. He's very active in clinical trials as a biostatistician. He's actually very engaged in national programs for development of the best biostatistical approaches to clinical research trials. There's an Association of Clinical and Translational Science that has an annual meeting that some of you may want to attend as you go forward. It's called the Translational Science Meeting. It's this year, I think it's in early March, but in Washington. If you want information about that, we'll be glad to help you. It's a great meeting. And he's the chair of the Statistical Design Special Interest Group within that meeting. And he's very active in the consultative program that the CCTS offers in terms of helping make sure that you have statistical support for any trials you're trying to do. So thanks, Bob.
Because my time is short this morning, I'll be going through the slides quickly and just highlighting key things in them. I don't think you'll be missing that much by me not going through everything, because Mansoor actually already touched on some of these things. As you'll see, it's sometimes difficult to completely separate the statistical components of the trial from the scientific components and things that the primary investigator would be dealing with. So that they're all, I think what I'm trying to say is they're all closely related and everything is intertwined. I'll just be speaking specifically about phase 1 and phase 2 trials. I know Mansoor mentioned a few things about phase 3 trials. If I were to speak on those, I could use an entire lecture just to talk about those. And I think that some of the concepts in those, though, you'll be getting later in the course. I believe that you have two sessions that are coming up in the future on biostats. And so some of the concepts, such as randomization, blinding, stratification, crossover designs, those should be covered in there. I'm always happy to answer any questions you have on those, but I'm not going to focus on those concepts in this talk. And my slides are intentionally dense. I'm not planning on reading everything to you, but I wanted you to have something to take with you after the talk that you could refer back to. And hopefully having this slide set plus Mansoor's will give you a bit of information about these types of clinical trials that you can use in the future. And I think that he already said some of these things about phase 1 studies, but from I guess the statistical aspects, most phase 1 studies are based on one of two common dose finding methods, but there's many variations to each of those. And I think we've seen almost all possible variations in our Cancer Center Protocol Review Committee. And I'll talk a little bit more about those in a minute. As I think Mansoor said, phase 1 trials are by their nature multi-center just because of the small number of patients who would be eligible and willing to participate in these trials. Phase 1 studies are considered hypothesis generating. So at the bottom, I put what phase 1 studies are not. Again, some of these concepts are not blinded, they're not randomized, and they're not used to test hypotheses. From my viewpoint, phase 1 studies are also not statistically sophisticated in terms of statistical analysis. The statistical sophistication comes in the design part of the phase 1 studies. There's 3 major types of phase 1 studies as I've laid out. There's role-based designs, and those are really the ones that have been in most use historically, and they're still in wide use today. Those are fixed designs where everything's laid out neatly. And once the design is set, the investigators essentially follow the rules of those of that type of design. I'm making that statement for a reason. The second one are pharmacokinetically guided dose escalation designs. These are actually not used very often, and I'll talk more about that in a minute too. And then the third one are adaptive model-based designs, and these are the ones where the design, we start out with the design, but the characteristics of the design can change potentially after each patient is enrolled and then evaluated on the treatment. And so in this type of design, this one requires, well, all of these designs require input from a biostatistician up front, but the adaptive designs require considerable biostatistical support as the study is ongoing. So if you do use this type of design, make sure that you have a biostatistician who, well, one who would have time to do this, but also who would be available and easily available while the study is ongoing. And an example of the rule-based designs are the 3+3 designs and accelerated titration designs, which Mansoor talked about. So those are things I'm just going to touch on very briefly. For the adaptive model-based designs, many of those are the continuous reassessment methods, and then the various modifications made to those. So the 3+3 dose escalation design, I've written everything out here for you. So I don't think I need to say a lot about it since that was already discussed by Mansoor. I will say though, to understand phase 1 designs and phase 1 studies in general, you do need to have a good understanding of this design. This is the one that's been used for, well, probably before I was born, actually, is when they started using it. So it's been used for many years historically, and it's still used, and it's still a very useful type of phase 1 design. And it may not be ideal. In fact, it's not ideal for all to assess all types of new drugs that are coming out now, and may not be ideal for all patient groups, but it still is ideal for many of both of those. The nice thing about the 3+3 design is it's just laid out very neatly, and you'll have rules that tell you when to escalate the dose, when not to escalate the dose, and those are based on the dose-limiting toxicities of the patients. I will say in some, there may be, for a phase 1 study, I've seen them and I've helped design these where we may have been just looking at as few as two dose cohorts, and some we may be looking at 6 or more. I've seen some from pharmaceutical companies that had as many as 10. I don't think they actually expected to escalate and to test all 10 of those cohorts, but they planned for that in case the lower doses were not effective in the patients. And I wrote out the most common definition of an MTD, and I don't remember if Mansoor gave you one in his handout or not. If he did, it should be similar to this one. And once we get through the phase 1 design, often an additional cohort of 6 or more patients will be enrolled at the MTD to further test the drug for safety. So when I'm evaluating phase 1 designs, the statistical parts of those, I look for some discussion about well, in addition to the number of cohorts, I look for some discussion about how many patients are expected to be enrolled. Now with the 3+3 design, it's pretty easy to figure out the minimum or maximum number of patients. The minimum number of patients will always be 2. And that's because according to 3+3 design, if the first 2 patients have DLTs, I mean, we're not going to escalate the dose, and we're not going to continue the study. I mean, the study will most likely just be shut down at that point because the drug is so toxic. So how do we determine the maximum number of patients? Well, I wrote that out down in the next step. And that's essentially, even though we're fairly certain when we design these, we won't have 6 in each dosing cohort, we should plan for that as like a worst case scenario. So the maximum number of patients in the study would be, since we can have 6 in a cohort, it'd be 6 times the number of dose cohorts we plan to examine, plus any additional patients that we plan to enroll at the NTD to further test the drug for safety. So that's the maximum number we would have on a phase 1 study using a 3+3 design. The accelerated titration design, which Mansur also mentioned, this is really it's a modified 3+3 design. The primary difference is that it allows for very large dose increments. So instead of allowing for, I'm just kind of throwing this number out, like a 5 or 10% increase in the dose, it may allow for a 30 or 40% increase, with the thought being that the lowest dose is almost certainly not going to be effective in the patient, but that it still needs to be tested regardless. And this type of design, there's statistical probabilities that I could calculate in terms of like what's the probability of dose escalation given the dose increment that we are using. And then the last design I mentioned, those are based primarily off of animal studies, and the starting dose is actually based on doses that were tested in animals. So it's that one's kind of a tricky one for, I think, even for an investigator to set up, and many assumptions need to be made about that. Probably the primary one is that the effect that was noticed in the animals at that dose will be similar to that noticed in humans at a similar dose, or a dose, say, that can be extrapolated, easily extrapolated from the animal data. Now, the continuous reassessment method, I wrote increasingly popular. That's been more popular in recent years. But that may have been more true a few years ago. I've noticed in recent years that although these adaptive designs, of course, are used, their use hasn't been increasing like exponentially, like it seemed to be a few years ago. And I think that's because there's some issues with the adaptive type of designs that might be viewed as undesirable. One of those is my second bullet point there, in that that when we design these studies, we may only have a single patient in some of the dose cohorts. And I've never met a physician or any investigator who's really comfortable with looking at a dose in only one patient. Obviously, every patient is different, and there's a very limited amount, the information that you can get from just one patient will be on a drug is limited. But this type, and a truly adaptive design, though, it allows for that if you're testing a drug and there's no the patient has no DLTs or there's no effect notice, and you just escalate the dose and go, and then go to the next patient and use a higher dose. So is that ethical or is it safe? And those are questions that have to be answered when this type of design is being set up. Now, the models used to set up these designs are based on Bayesian principles, so a biostatistician is very involved in setting up this type of design, and really no two adaptive designs are quite the same. And so, but the two issues are, like I said, the single patient cohorts. It's possible to have those. The other issue with this is that using this design may overestimate the dose for the MTD, and that's because not enough patients were studied at the lower doses. So that's the trade-off with using an adaptive design versus a fixed one. So how have how have investigators gotten around that? I mean, well, they work with statisticians and come up with modified adaptive designs. And these designs incorporate traditional designs such as aspects of 3+3 designs, along with some aspects of adaptive designs. And the idea is to improve upon the patient safety of the study, and also to arrive at results from these studies that might be more convincing to folks. So for example, it's possible to set up an adaptive design, but to set a lower limit on the number of patients that will be in each cohort. So the number in each cohort still may vary, but one might say, well, we have to study at least 3 in each, but the maximum might be 6 or it might be 10. I mean, we don't know how many will be in higher dosing cohorts. And there's something brand new here, and something I couldn't answer questions on now because it's so new. But there are 2-stage likelihood-based CRMs being developed now, and I tried to look that up some on the internet and other places, and one of the things that I found was a graduate student's dissertation based on these. So that's how new these are. And I already talked about the first point here. So when I'm evaluating these studies, that's one thing I look for, is justification of the sample size based primarily on the number of, well, based on the designs that 3+3 are adaptive, and then also on the number of dosing cohorts. Another way to justify phase 1 designs are what we call using confidence interval approach. Or I should say an additional way. And this doesn't replace the first one. But if we have a hypothesized toxicity rate, which we often do in phase 1 studies, then we can calculate a confidence interval for our expected sample size. So once we go through the design, I gave examples for sample sizes of 10 and 12, and we may end up having a phase 1 study with more. We could have like 20, but we're not going to have like 100 patients in a phase 1 study, right? They're going to be small studies. But, as you can see, though, by those results that I gave you, the width of the confidence intervals are going to be very wide for these small groups of patients. So, I mean, so this adds more statistical weight to the protocol, but in terms of practical information, I'm not sure how much practical information it adds, because we already know before calculating this that we're going to have very wide confidence intervals due to the small patient sizes. Now, for data analysis methods on phase 1 studies, this is the part I said would not be statistically sophisticated. These methods are primarily descriptive. So descriptive statistics, they're important in every study, but they're even more important in phase 1 studies because we may not be able to do a lot of other analyses than that. We can calculate confidence intervals for toxicity rates or any other rates that the investigator is interested in. In the rare cases where we do have, say, multiple dosing cohorts where we ended up with 5 or 6 patients in those, then we can statistically compare those using conventional statistical methods, such as analysis of variance. I seldom see phase 1 studies where we have enough patients that we can actually compare dose cohorts that way, but it does happen once in a while. And it may be feasible. The reason I put the fourth point in is because I was involved in a study where we did just that. We were able, it made sense in terms of the science to combine data from lower dose cohorts and then the data from higher dose cohorts, and then perform a low dose versus high dose comparison of the drug. So that's one thing that we might be able to do statistically. And the statistical tests, again, they'll be limited to descriptor statistics, maybe analysis of variance, maybe the two-group t-test, and the appropriate non-parametric analogs to those. But that's really all the statistics we can do with the phase 1 study. Oops. So I'll go ahead and move on to phase 2 studies. One thing I didn't show, but maybe I should have. Okay, maybe I should have given an example. For phase 1 studies, biostatisticians often do calculate the probabilities of dose escalation in those. So that, again, that's based on patient numbers and based on expected toxicity rates. So we have to start with somewhere, but sometimes we do create tables of those in the protocols. So that's where statistics is involved in phase 1 studies, in those aspects. For phase 2 studies, I've outlined some things. Again, some of this is just descriptive about phase 2 studies. But in terms of statistics, though, we may just have one group in the study. We may have 2 groups in a phase 2 study. We actually could have more than 2 groups if one or more of those groups is a control group. I mean, I've seen all these types of phase 2 studies done before. If we only have a single group, though, we usually compare the pre-treatment status versus the post-treatment status of those patients. Because we're not going to do a study unless we can have some type of comparison involved. So I would say most of the time we'll compare, like, say, a treatment group versus a control group. Multiple doses could still be examined in a phase 2 study, but typically each dose would involve its own patient group. So we wouldn't do any dose escalations in a phase 2 study. Now designing a good phase 2 study depends on having good phase 1 data to help design the phase 2 study with. So that's very important. Phase 2 studies are also used then, well, I should say maybe used to design phase 3 studies later on. It depends on how the results of the phase 2 study comes out. I don't think I mentioned this before, but phase 3 trials are the ones that can involve hundreds or maybe even thousands of patients or participants. So, but before doing a phase 3 trial, we want to have some excellent results from our phase 2 studies. Phase 2 studies may consist of either 1 or 2 stages, and they may have 1 or multiple arms in them, and the arms are not necessarily the same as the stages. I'll explain a little bit more about the stages in a minute. Mansour mentioned stratification. Well, in phase 2 studies, we may stratify those studies according to various clinical and demographic factors. What has kind of fueled the growth of 2-stage phase 2 studies are an increase in computing power because to design those, we need the, well, it's based on probabilities, but we need computers to help us calculate the probabilities and the design characteristics. And up until the mid-1980s, statistical research is very limited on phase 2 studies, and there is very little about two-stage designs. Most of it was just focused on single-stage phase 2 studies. So when I say a one-stage, refer to a one-stage phase 2 design, the sample size is fixed, and there's not a conditional patient group or a conditional outcome that we're looking at. We just design the study, do the study as planned, and then do the data analysis. And that's, I mean, that's that. In a two-stage design, the second stage is always conditional on the results of the first stage. So when we set this up, we do need to plan, or we should plan for the maximum number of patients that could be enrolled in both stages, knowing that we may never actually carry out the second stage. The purpose of the first stage is to rule out drug or treatments that have little or no biologic activity. And that's usually set by the investigator, and the investigator would have good ways to justify why he or she chose that level as the minimum activity level. And so, for example, if the estimated if it was set at 20% initially, and if after the first stage, once we look at all the data and determine the activity level is less than that, then we would not advance to the second stage. We would just end the study right there and report the results. Now, in many oncology studies, I think that patients who are enrolled in those often don't have many treatment choices. So the bar is set low in that the minimum level of activity in the first stage might be as low as 5%. So basically, if the physician sees promising results in even just 1 or 2 patients, then they may want to move on to a second stage and examine more patients with that drug. Now, even though if we proceed to the second stage, we'll have patients that are in each stage. But when we do the analysis, we consider it as one study. So the response rate for the study is based on the total number of participants who were enrolled in both stages. So, and the last point is, the sample size for each stage is fixed in advance, and that's based on the statistical design and on probabilities. Now, in terms of sample size justification, for a one-stage design, since there's not a conditional stage or conditional outcome, the justification for sample size for those can be, well, it is based on conventional statistical methods for 1 and 2 groups. And I know that you'll be talking more about those in your future lectures on biostats. But those would be ones that you've heard of or ones that you've read about that are very commonly used. Oh, I should have mentioned that in phase 2 studies, the variables can take on different forms. So the outcomes can be the full range. They might be continuous variables. They're measured on a continuum, or they're categorical, such as a binary variable, or they might be some type of survival outcome. All of those outcome variables are permitted in phase 2 studies. Now, moving on to the third point. Okay, I'll try to go a little faster here and get done. For two-stage designs, up until the 1980s, EHAN designs were the standard ones used. Those aren't, well, I seldom see those, and I would say those designs should not be used anymore. In fact, if I see one in a protocol, I always question it. In 1989, Richard Simon published a seminal, and which has now been a very well-cited paper about optimal stage 2 designs for phase 2 trials. And that's become the standard for all two-stage phase 2 designs from that point on, even to the present day. Now, there's been other statistical research done and some modifications made of those, but they all come back to what we refer to as Simon's two-stage designs. So if you're reviewing a protocol or setting up a phase 2 design, and it's one that you think might need 2 stages, then we would want to use a Simon's two-stage design for that. And those designs, again, are based on various probabilities. And we do need statistical software to help us set up those designs and to determine how many patients should be in each stage. So those aren't calculations that are easy to do by hand. In terms of data analysis for phase 2 studies, again, we would compute descriptive stats. We can't, though we can use several conventional statistical methods for group comparisons where that's appropriate for the phase 2 study. And where we can analyze, I should say, pre-treatment versus post-treatment, we can use statistical methods designed for paired data. That would be such as a paired t-test or Wilcoxon sign rank test. If our outcome is a survival outcome, we can apply survival analysis methods such as the Kaplan-Meier method and the log rank test as well. So there's much more we can do statistically in terms of analysis for data from phase 2 studies. Now the question might come up, well, we did a phase 2 study and we ended up with a small number of patients. Well, that may limit some of these methods that I've listed here. It may limit the use of some of these methods. For example, the survival analysis methods, you do need, I don't want to say there's a set minimum number, but if we only have a handful of patients, trying to run survival analysis on those is not going to produce meaningful results. Actually, I think this is my last slide here. And I really would have liked to have included more or talked more about some of the statistical aspects and detail of these studies, but I know that during this course, my time is limited for that. In fact, all the speakers you have, they just have a limited amount of time for their topics. But I think in every talk I give, I always have one slide on software. I think it's important to mention something about that. The software that statisticians would view as being most definitive in terms of sample size and power calculations are nQuery and Visor plus Interim. Sometimes we just refer to that as nQuery, but that's just one package. And the other is PASS. And the reason is both of those are very complete in terms of the variety of sample size computations that can be performed. Now, there's many other web-based software, some are free, but most of those can only perform calculations for just a very limited for very limited situations. So they may be able to perform calculations, just say if you're comparing 2 groups, or comparing maybe one group and you want to do a pre versus post comparison. But that might be the extent of what the online ones can do. And they won't be able to take into account other factors that go into these, well, that may be part of your own studies. The second bullet point, I list software that are most commonly used to perform statistical analyses. And I'm not implying that other ones out there aren't good or that they're not accurate. I know firsthand that there's many physicians who will start doing some of their own analyses using Prism. That's a popular software package. It's very easy to use and it's menu driven, but it doesn't have the more advanced statistical methods as part of the software. Whereas in these packages here, these are very complete, and so, but even among these, some require more learning than others. I would say all of them require some learning. None of these, I don't think, you can't just sit down and use like after looking at them for 5 or 10 minutes. But like, for example, SPSS is mostly point and click, and much of Stata, I think, is point and click mode. You can still write some code for both if you choose to. Whereas R and SAS, those may take a year or more to become proficient with those software packages. And in fact, there's classes offered here, even here at UAB, on learning to use SAS, for example. There's semester-long classes. There may be one on learning to use R now, too. I'm not sure. I'd have to ask my colleagues in the biostat department about that. As investigators, you would normally not be expected to do all of your own statistical analyses. So I actually like it when investigators do some of their own preliminary analyses, even if it's not optimal and even if they don't use great software, because then they actually get I think, a better feel for their own data before coming to me, and I think it makes our discussions and collaborations more focused. I think I've said everything here. Oh, with computers being the way that they are now in terms of hardware, RAM and processors and so on, they're really not an issue these days with modern computers. I think that they can run any of these software packages on laptops or on servers or on mainframes. I guess for those who still use mainframes to run their analyses on. And so as long as you're using a modern computer, one that was built in the last few years, I think any of these software packages will run on them. And then the last slide is just a few key references here. There's actually many references. I would say there's many statistical references on phase 1 studies and also on phase 2 studies. And I could give you a more extensive list if you would want that.